I hope you all had a pleasant lunch break. So we shall now proceed with the oral presentation sessions. So we have TCO 19 to TCO 23 and TDO 24 to TDO 25. I repeat, TCO 19 to TCO 23 and TDO 24 to TDO 25. And I request all the presenters to kindly remain ready according to the schedule and please adhere to the time. So now I request the chairperson and co-chairperson of this session to kindly proceed with the proceedings.
Okay, thank you. Welcome back, all of you. Good afternoon once again. So, the oral presentation for this technical session began with the first person is Sadat, and his topic is nest site selection and breeding ecology of Brahmini kite in Shimoga, Karnataka, India. And I request all the presenters, please stick on to your timings. So, as we are running out of time, please present your time only within 4 minutes.
Good afternoon, everyone. In the morning session, we had a discussion about the mammals as well as the amphibians. Afternoon session will be from my side. It will be from birds, aves. So, I am Dr. Sadat Alias, Assistant Professor, Department of Zoology, Khaja Banda Nawaz University. Today, I am going to present a small part of my PhD work, that is nest site selection and the breeding ecology of Brahmini kite in Shimoga. Coming to the introduction, the Brahmini kite is a conspicuous raptor with a large range in the Indian subcontinent, Southeast Asia as well as the Australia. Brahmini kite has a white head and reddish brown colored body. They are mainly found in the rivers, wetlands and the agrarian fields. They primarily feeds on fishes and other prey items. Breeding season of the Brahmini kite is from December to April, and the last batch of the fledgling leaves the nest in the month of May to June during the onset of monsoon. They show considerable site fidelity. That means they nest in the same area year after year. These are the materials and methods. Study area. The study was conducted across the sampling sites in Shimoga Taluk. The landscape is predominantly characterized by the agrarian fields interspersed with the riverine habitats. Coming to the methods, active nests of the Brahmini kite were located by searching their known territories and observing the soaring birds. The nests were observed during the regular brief visits. Focal animal sampling technique was employed to monitor the parental care as well as the activities of the nestlings. The observation spots were always placed within the 100 meter of the iris with caution so as to not to disturb the breeding pairs. Coming to the results and discussion, breeding season of the Brahmini kite in India is from December to April. The nesting began in the month of December, and the nests were found in the agricultural areas as well as in the periphery of the water bodies. The nests were found on the Enterolobium salmon as well as the Ficus religiosa species. The mean nest height was 15 meter below the ground level and 1.7 meter from the apex of the tree. And this is the table, the nest site characteristics and the breeding parameters recorded in the different habitats of Shimoga. You can see here So usually the Brahmini kite builds two nests. It builds more than one nest and it uses another nest as a backup. It stores the food materials, such as if it hunts a larger mammal or a larger bird, it will store the nest in the backup nest. It will store the food. And when it is required, it uses the food from these nests and feeds the chicks. I have a video of that, I will show you. And the incubation period of 26 to 28 days were observed and the nestling time was 44 to 48 days. And you can see here, this is nothing but an ontogenic development. There is nothing like a camouflage. This is the juvenile Brahmini kite. You can see the contrast between the adult as well as the juvenile. So the juvenile body is completely dark brown in color, whereas the adult Brahmini kite, you can see here, the adult Brahmini kite has a white head, whereas the juvenile has completely a dark brown in color. As it ages, as it becomes sexually mature, the coloration of the body changes. And this is the prey composition. The majority of the prey composition was found to be fishes, which is followed by arthropods, amphibians. And the nest was built near the agricultural field for the easy availability of the food material. This is the diurnal pattern. I have also observed the diurnal pattern of the prey delivery, means at what time it delivers the maximum amount of food. It was found that on the morning hour, that is 9, 11, and in the noon, that is 4 o'clock, it delivered more amount of food to the nest. And you can see here, this is the Brahmini kite. It cleans the food, whether it hunts fishes or arthropods. It first cleans the food and then it feeds its chicks. And there is another video here. See here, this is the backup nest. You can see a house sparrow. This is a bird. Actually, you can see here, this is the bird it has hunted and it has placed in a backup nest. When the food is shortage, it uses the food from the another nest. And coming to the summary, the Brahmini kite is a synanthropic species which is widely distributed and occurs across the Indian subcontinent. Breeding season is from December to April and the incubation period of 26 to 28 days and the nestling period of 44 to 48 days were observed. And the prey composition was dominated with the fishes, followed by arthropods, amphibians, and the diurnal pattern was also observed, the peak delivery of the food. This study demonstrated that the Brahmini kite successfully exploited the heterogeneous agro-riverine ecosystem by flexible nest size selection, opportunistic feeding, and temporally optimized prey provision activities. So these are my findings and these are the references, and thank you.
Thank you, Sahyadri. Any questions? Any quick questions from audience?
Yes, sir.
Okay.
Yes, sir.
It's a wonderful.
Thank you, sir. Thank you.
Yeah. Actually, I had one doubt. ಏನಂದ್ರೆ ಈ ಫಿಶ್ ಹಿಡಿಯುತ್ತೆ ಅಂತ ಹೇಳ್ತಾ ಇದ್ರಲ್ಲ.
ಹೌದು ಸರ್, ಹೌದು.
Is there any specific fish ಏನನ ಇದೆಯಾ ಅಂತ?
There is no specific fish, sir, but as it lies in the bank, actually I found the nest near the Javali, sir. It's in the bank of the Tunga river.
Okay.
So there, based on the availability of the fish, it eats, but it is not specified to a particular fish species, sir.
Nothing else.
Yes, sir.
ಯಾಕಂದ್ರೆ ಬ್ರಾಹ್ಮಣಿ ಕೈಟ್ ಹಾರಾಡ್ತಾ ಇದೆ ಅಂತ ಹೇಳಿದ್ರೆ
Yes, sir.
ಅಲ್ಲಿ ಈ ತರದೊಂದು ಏನಾದರೂ ಒಂದು ಫಿಶ್ ಇದೆಯಾ ಅನ್ನೋದೊಂದು ಐ ಹ್ಯಾಡ್ ಅ hypothesis.
Yes, sir. Yes, sir. Definitely, yes, sir.
Thank you.
No, sir, there is no such specificity, but it feeds majorly on different kinds of fishes, sir.
Thank you.
So you mentioned that the bird stores its food in the backup nest
Yes.
when it uses when the food is insufficient.
Yes.
So what happens when there is sufficient food and it has like enough stored food in its backup nest? Does it eat the backup stored food first, or does it freshly hunt?
As like humans, they usually prefer the fresh food, but not in such cases. But usually when the food is shortage, or when actually during the month of April, the flow of river will be in short. So they usually store the food in that places because the nestling time was observed during the month of April to May. During this time, the fishes availability is very less. So it will usually hunt the mammals as well the birds, arthropods. If the fish availability is more, they primarily go for the fishes. Their first choice is fish. If the fish is not available, they go for arthropods, amphibians, mammals, and the small birds. So this is how it adapts its feeding behavior.
Okay, thank you, Sahyadri.
Yes, sir. Thank you, sir.
Thank you. So the next presenter is Aparna, and our topic is colorimetric lamp for rapid field deployable molecular wildlife identification.
ಇದೊಂದು
Good afternoon, everyone. So I'm going to today talk about a futuristic technology called LAMP assay. So this is in context with the current theme of emerging trends in technology in conservation and management. So I'm going to talk about the colorimetric lamp for rapid field deployable molecular identification method. So DNA based identification is central to So DNA-based identification is central to a lot of decision making for various types of on-spot decision making. So me coming from the background of forensics, my lot of focus has been on identifying species in the context of difficult samples where their morphological identification has been very challenging. Apart from that, DNA is very helpful in identifying the species, and although there are not many studies in terms of having databases to affiliate with its geographic origin in terms of widely distributed animals, DNA based identification is still holding the key to a lot of decision making processes. And definitive evidences through these scientific approaches help in further decision making in terms of the legalities of it. Now, there is a need for rapid field based identification because when we talk of DNA, the processes are quite laborious and largely lab based, which may take between few weeks to few months, and there's a lot of costing also involved when these type of tests have to be done. So field level dependency, lab related dependency is more, and we are trying to, with this technology, bring it to the field so that analysis can be done on spot. So just a brief about LAMP assay. This is abbreviated as loop mediated isothermal amplification. So as the name indicates, isothermal amplification means the DNA gets amplified at a single temperature and multiple copies are generated. So how this is better over the traditional methods are that the same kind of a readout is obtained, but the readout is much better and quite quick and much cost effective also. The average results are obtained roughly between 30 to 45 minutes, which is very useful in field conditions. And it also eliminates the need for thermocyclers or advanced laboratory infrastructure. The readout is a visual color change or a turbidimetric change or a fluorescence change, as we can see here, comparative to PCR-based methods, where there is a need for instrumentation and a dependency upon the complex data that is generated through it. But in terms of a color change, it is very easy to read, and anybody can be trained in order to interpret the results associated. On the basis of this, since there's a lot of gap between the procedural aspects, this proposed technique can be a very useful tool for field deployable molecular based identification. And this has a capability to analyze a diverse range of samples, because we get many types of samples like tissues or degraded samples, process derivatives, and they can also be derived from both plant and animal based sources. So in several reports like this, DNA traces at livestock, it can help in better identification of which type of wildlife can be associated with it. These type of technologies can give on-spot decisions. And these are some focus areas. It can help in identifying conflict animals. It can help us differentiate between protected, endangered, or common species. Geographic source identification and population dynamics can be explored. And also information pertaining to whether they're wild sourced or captive bred can be identified on spot. These are some portable instruments for molecular methods called Bentolab and a mini ion sequencer. So this can just simply be carried to the field and the data can be generated and decisions can be made as such. So this is the first of its kind of study in India. And like I said, a lot of benefits associated with rapid testing. And this is a confirmatory test on spot because it's based on a DNA identification. And this can also be integrated to certain devices like microfluidic devices so that officers can be trained in order to identify what the associated species are. So this is a greener, most cost-effective and time-effective method. Some of my acknowledgements. Today also happens to be Endangered Species Day. So futuristic technologies can be, like this can be implemented on field and it will help various conservationists and management of wildlife better. Thank you.
Thank you, Aparna. Any questions? So I have my own questions, Aparna. First, yes. I will ask the question. So is there any chance of contamination in field studies? How to mitigate that?
Yes, contamination-based study, it is possible. But it's also, this technique is highly specific because it's a molecular method and there are specific markers associated. So we need to kind of reduce the source of contamination if possible. But even otherwise, the limit of detection is up to 10 picograms, if I'm not wrong. So since the sensitivity is very high, even in the presence of contamination, it can still pick up specificity for identification. So that is definitely possible.
Thank you. Any questions? Okay, thank you. If no questions, thank you, Aparna. And the next person is okay, Sahyadri. His topic is dietary influences on gut microbiota of wild sloth bears in Jassur Wildlife Sanctuary, Gujarat.